Welcome to The Deep Dive, the show that extracts the crucial knowledge from complex research, giving you a shortcut to being well-informed.
Today, we are bridging what used to be, well, a massive gap between clinical risk and what's actually happening at a cellular level.
Exactly. We all know the high-level fact. Chronic hypertension, high blood pressure, it increases your risk of cognitive decline as you age. That's a well-established medical connection. But the question that has, really stumped researchers for a long time is, what specifically is breaking in the brain's delicate memory hardware?
And we're not talking about general wear and tear here.
No, not at all. We're talking about the functional electrical circuits.
Right.
Our mission in this deep dive is to connect that broad clinical correlation directly to a specific neural circuitry breakdown based on some really compelling evidence published by Ratner and colleagues in 2025.
Okay, let's unpack this.
That's right. The clinical data is showing that if you therapeutically control hypertension, it reduces cognitive risk. That's solid. But it's a big picture systemic intervention.
It is. What this research does is it zeroes in on the microscopic, the underlying functional changes in the actual neural circuits that dictate memory consolidation.
So the goal is, if we can pinpoint exactly where the functional machinery is failing.
Then we move beyond just managing blood pressure. We start identifying potentially new therapeutic targets right inside the memory center itself.
Which means looking deep inside the hippocampal trisynaptic circuit.
That's the one, the HTC.
Ah, the HTC. We hear that acronym a lot, but for our listeners, let's just clarify why this circuit is so fundamentally important to memory. Why is it the target here?
Well, you can think of the HTC as the brain's internal filing system, specifically for transforming our immediate experiences into lasting memory.
Okay.
It's a three-stage pathway. Information comes in, it gets processed, and then it's ultimately funneled through the CA1 subregion of the hippocampus.
And the CA1 area is the final output, isn't it? The crucial bottleneck.
It's the bottleneck,
Yeah.
exactly. It's where memory either gets successfully consolidated or it just, it fades away. If this area is dysfunctional, your memory system is failing, period.
And the need for this kind of mechanistic understanding, it's really driven home by just the sheer scale of the clinical findings that prompted
No,
this.
absolutely.
We're talking about massive studies. Here's one cited here that tracked, what was it, over 500,000 participants?
502,537 people, yeah. And it showed this really powerful relationship between a reduced prospective memory score and, critically, reduced hippocampal functional connectivity in patients with a history of chronic hypertension.
The correlation is it's undeniable.
It absolutely validates the need to move from that correlation to the actual mechanism. And the way they measured the health of the CA1 bottleneck wasn't just structural, but functional.
Using an electrical biomarker, the short wave ripples, or SPWRs.
That's it.
So tell us a bit more about these ripples. What are they, functionally speaking?
They are synchronous high-frequency oscillations. They happen between about 140 and 200 hertz, and they occur during moments of quiet wakefulness or immobility.
So when you're just resting.
Exactly. You can sort of visualize them as these intense, rapid packets of electrical activity. It's essentially the system replaying and consolidating historical information.
Like downloading data into long-term storage.
It's a great analogy. The ripples are the physical manifestation of that process. If your ripple amplitude is strong and clear, consolidation is happening robustly. If they're quiet and weak, well, the filing system is impaired.
Okay, so to study this link in a controlled environment, the researchers turned to an animal model known to naturally develop this problem, the male sprog dolly, or SD rat.
Right. SD rats are a great preclinical parallel because they reliably develop increased mean arterial pressure, or MAP, and they exhibit measurable memory impairments as they reach advanced age, so around 16 months.
So it's a model that really mimics age-associated hypertension and its cognitive consequences in humans.
It gives them a perfect window into the process.
So we have the circuit, the HTC, and specifically CA1. We have the biomarker, the ripples, and we have the subject, the aged hypertensive rats. Now, how did they test the functional integrity? They didn't just observe the ripples, did they?
No, they stressed the system. They used a chemical tool, a nootropic drug called I5IA.
A probe drug.
Exactly. I5IA acts as a highly specialized probe. It lets them test the capacity of the circuit to respond to potentiation. Its action is very specific. It selectively inhibits a 5-GABA A receptors.
Okay, wait. GABA is the main inhibitory neurotransmitter. So this drug By blocking certain receptors, it reduces inhibition, right? That should generally make the circuit more active.
Precisely. But here's the crucial mechanistic nuance. These I5 GABA A receptors, they mediate two types of inhibition in the hippocampus, tonic and phasic.
Okay.
Phasic inhibition is, quick decisive signaling, like a specific command or traffic light turning red. But tonic inhibition, that's the persistent low-level background conductance.
Like a dimmer switch.
It's exactly like a dimmer switch, always controlling the background excitability of the entire circuit.
So I5IA messes with the dimmer switch.
It does. It selectively turns down that tonic background inhibition. Now, in a young, healthy circuit, when you relieve that background inhibition, the circuit gets revved up. It potentiates.
So you see a big increase in the peak ripple amplitude.
A significant increase, yes. If the circuit is functionally intact, this drug should crank up the volume of those memory ripples. It's a gold standard test for the health of that circuit.
This sounds like a very rigorous experimental setup. A within-subject design, probes implanted in awake, freely behaving rats, administering the drug orally and measuring the ripple response as the dose increased.
And we really can't overlook the critical importance of their control group comparison. They contrasted the hypertensive SD rats with another strain, the Fisher 344 or F344 male rats.
Right, and why are the F344 such a good control?
Yeah.
If aging causes a generalized decline in brain health, wouldn't they still show some ripple decline at 16 or 18 months?
They might show some general aging effects, perhaps, but F344 rats are genetically not prone to developing age-related hypertension.
Ah, so that lets them isolate the variable.
It does. Previous studies had already shown that F344 rats retained a completely robust, positive ripple response to this F5IA challenge, even when they were quite old.
And that's the key piece of evidence, isn't it? The resilience of the ripple mechanism in those F344s proves that aging alone isn't the primary destructive factor here.
It strongly suggests it's the hypertension associated with the aging process that is actively disrupting this memory circuitry.
Okay, so here's where it gets really interesting. When the researchers introduced the I5IA probe to the aged hypertensive SD rats, the circuit had fundamentally changed. They found two major anomalies.
The first one was just baseline weakness. Before they even gave the drug, the aged hypertensive SD rats showed a measurable reduction in their mean peak ripple amplitude compared to the young, healthy rats.
So the high-amplitude ripples, the most effective memory signals, were already reduced.
They were. The researchers noted what they called a distinct left shift in the frequency distribution, which just means the average memory packet was already quieter.
Their memory consolidation process was running on a lower battery to begin with.
That's a good way to put it.
But the second anomaly, the real smoking gun about functional integrity, was the response to the challenge. That expected potentiation. The increase in ripple volume.
It was significantly reduced or lost entirely. The circuit failed the stress test.
It just couldn't respond.
It lost the capacity to respond to the pharmacological manipulation. In a healthy system, you relieve the inhibitory brake, the engine should rev.
Right.
In the aged hypertensive rats, the engine was fundamentally broken. It wouldn't accelerate. And this indicates a pathology that's specific to that GABA A receptor or its downstream signaling components.
And they could perfectly quantify this failure, right? They classified, 50% of the aged SD rats as outright non-responders?
Half of them. And crucially, they weren't guessing about the cause of this non-response. It was directly tied to blood pressure.
So the non-response was statistically associated with mean arterial pressures over 160 millimole Ricci.
Yes, and this is maybe the most significant functional link they established. They performed a linear regression analysis and found this powerful inverse correlation.
Let's make that technical detail accessible for you. They reported an R-squared value of 0.86, which is, that's incredibly strong. What does that practically mean?
It means the relationship is nearly linear and almost perfectly predictive. Imagine plotting a graph where the horizontal axis is the rat's measured blood pressure, the MAP, and the vertical axis is the circuit's ability to respond to the drug.
Okay.
An R-squared of 0.86 means that as the blood pressure goes up, the circuit's functional responsiveness goes straight down, almost in a perfect inverse line.
Wow.
The severity of the hypertension correlates directly and powerfully with the severity of the functional deficit in the memory circuit.
That pretty much removes all doubt about the connection. And just to confirm that this electrical failure translated to a real-world impact, they backed it up with behavior tests.
They did. Separate groups of the same 16-month-old hypertensive SD rats performed significantly worse on delayed recall tasks.
Specifically, the novel object recognition test and the location novelty recognition test.
That's right. The behavioral impairment in their memory aligned perfectly with the electrophysiological observation that their memory circuits were functionally broken, unresponsive.
So if we connect this all back to the bigger picture,
This research provides the first mechanistic demonstration that age-associated hypertension doesn't just cause some diffuse general brain damage. It specifically targets and compromises the signaling architecture, these sharp wave ripples that the hippocampus uses for memory consolidation.
that completely reframes the discussion about cognitive decline in hypertensive patients. And suddenly, these high-frequency oscillations in the hippocampus, they become a critical, valuable, and potentially translational biomarker.
They absolutely do. This research suggests that a modified 5IA probe challenge could, potentially be developed into a novel objective diagnostic tool.
For detecting early memory deficits in hypertensive patients, long before the clinical symptoms become severe.
Exactly. Now, the gold standard for measuring these ripples right now involves implanted electrodes, which obviously isn't feasible in humans.
But the technology is moving so fast.
It is. The researchers themselves point out that non-invasive measurement of ripple activity using techniques like magnetoencephalography or MEG is an emerging possibility in human neuroscience.
So the ability to non-invasively test the functional response of the memory circuit, that offers a really clear path from bench research to a bedside diagnostic.
A very clear path.
Now, before we wrap up, it's critical to look at what the researchers themselves highlighted as limitations and next steps. For instance, the study established a powerful association, but not definitive causality, right?
That's the next frontier, yes. They've shown that high MAP and SERDIT dysfunction go hand in hand, but they haven't yet proven that actively lowering the MAP will restore the functional integrity of that circuit.
So we don't know if the hypertension is actively disrupting the CA1 receptors or if it's some secondary effect.
Correct. Future work has to determine if treating the hypertension can actually reverse or mitigate the circuit impairment and bring back that ripple response.
And the researchers also focused exclusively on the CA1 subregion, which is the final output.
Exactly. Since the ripples actually originate in a different region, CA3, and they're heavily shaped by inputs from the dentate gyrus and entorhinal cortex.
It's pretty likely that upstream dysfunction is also playing a role in altering the ripple architecture they saw in CA1.
Very likely. To fully understand the breakdown, they'll need to trace the dysfunction back through the entire trisynaptic pathway.
They also acknowledged a tight sample size for the electrophysiology part. I think it was 3 young and 4 aged rats.
And while that sounds small, the use of the rigorous within-subject design, where each rat served as its own control across the drug doses, that significantly maximizes the data reliability.
So it's challenging research, but that consistent high R-squared value suggests the foundational link is solid.
It does. It's very solid.
So the key takeaway for you listening is this. Age-related high blood pressure doesn't just damage your blood vessels. It fundamentally breaks a specific, crucial neural communication mechanism, the sharp wave ripples, that your hippocampus uses to consolidate memory.
And critically, this breakage renders the circuit unresponsive to certain types of pharmacological potentiation. The system is functionally corrupted.
It's not just weak, it's broken.
And since the profound loss of this I5IA responsiveness they saw in these hypertensive rats, since it mirrors similar findings in some genetic models of Alzheimer's disease, this raises a really important question. Does successfully treating age-related hypertension potentially offer a direct, measurable pathway, perhaps through early diagnoses using these ripples, to pharmacologically restore the functional integrity of these memory circuits?
Does he fix the blood pressure early enough?
Can we make the ripples loud again? Can we re-enable the mechanism for consolidation? That's something for you to mull or explore on your own.
That offers genuine hope for early intervention. That's it for this deep dive.